Hello, grade 10. So today we are going to complete our lesson manipulating DNA. And as we learned that the biotechnology relies on cutting DNA at specific places. And this is used after the DNA fragments being cut. These fragments will be used to be studied or to be moved from one organism to another organism for better characteristics. So the scientists use several techniques to manipulate DNA, and because DNA is very small, a very large molecule, but at the same time we cannot handle it by our hands, or we cannot see it because it's too small. Okay, therefore they use special tools to study DNA, such as computers, chemicals, and bacteria. All right. Now, chemicals are important to change the sequence of the nucleotides. Bacteria is important to get from it the enzymes that are used to cut the DNA at specific sites. Okay, and for slicing the chromosome into pieces or cutting it to pieces, we cannot use the normal scissor. We should use a molecular scissor, which are enzymes that we can get from the bacteria. So these restriction enzymes act as molecular scissors, and they come from various types of bacteria, and they allow the scientists to more easily study and manipulate genes, and to cut the DNA at specific nucleotide sequence called restriction sites. So each restriction enzyme can cut the DNA at specific restriction sites, and that means it can cut at a specific sequence of nucleotides, because each enzyme could recognize only specific sequence of nucleotides. All right, and we have different restriction enzymes that cut DNA in different ways. Each enzyme has a different restriction site. Okay, so as you see here, restriction enzyme 1 can cut this DNA strand through these restriction sites. He can recognize these restriction sites, which are specific nucleotide sequences, whereas the other type of enzyme can recognize different restriction sites. And here the fragments of DNA are less than here. We have 4 fragments as a result of the cutting, and we have here 7 fragments. And some enzymes cut the DNA with blunt ends. Okay, they cut straight across the leaf, and they have blunt ends, straight cut. And some make staggered cuts and leave sticky ends as these. And as it shows here, the sticky ends have free nucleotides, bases, and they are very useful in the biotechnology because they can stick easily with other segments of the DNA. Okay, and as you see here, this is a restriction enzyme called TAC1. It cuts DNA when it finds its restriction site, and its restriction site is this sequence of nucleotides, TCGA. It can recognize it in both ways, either this way or this way. If you recognize this site, this enzyme will start to cut the DNA as staggered cuts and leave the sticky ends. Now, after we cut the DNA fragments should be separated by using an gel electrophoresis, and this will show restriction maps at the end. So the restriction maps show the length of DNA fragments. We must separate the DNA fragments to see the length of each fragment. Okay. Now, the gel electrophoresis is used to separate DNA fragments by size. And okay, DNA sample is cut with restriction enzyme. It's ready. We put it in the electrophoresis. Electrophoresis is an electrical current pulls DNA fragment through a gel. Now, the length of DNA fragment can be estimated from the distance it travels through the gel at certain time. The smaller fragments move faster and travel farther than larger fragments. As you see here, these bands show how the fragments move. Like this band here, this band, it moves or travels farther from others. That means this fragment is smaller in size. Okay, so the bands here show a map, restriction map, okay, which shows the length of DNA fragments. So fragments of different size appears as bands on the gel, and as a result, the restriction map shows the length of DNA fragments between restriction sites. Okay. Now, it only in this technique, it only indicates the size, not the DNA sequence. This is very important to know. And you will know the length of the DNA segment, but it cannot indicate the sequence, remember the nucleotide sequence. And it is useful in genetic engineering. We are going to talk about it later in details. What is the most important you should know here? That restriction maps are very useful because they can study mutations. Okay, why? Because during the mutations, deletion or insertion may happen. Okay, in the nucleotides, and this will change the length of the DNA fragments. So they will compare these bands to a normal reference, and they will see if there is any change in the length of the fragments. That means there may be a mutation happened. Also, the mutation may change restriction site who a specific nucleotide sequence. And if this size is changed, okay, the DNA would not be cut because the enzyme cannot recognize this site, and they may find very long DNA fragments. Okay, so have a nice day and see you tomorrow. Take care.